Alright, yes, Assalamu alaikum everybody. How are you all? I hope that you all are doing well. Welcome to the class. Today we will be looking at AS and A level biology paper 1 2 of February March 2022, and this paper is of 1 hour and 15 minutes. So let's begin this paper with the question number 1 over here. Question number 1 over here says that which cell organelle are clearly visible when viewed with the light microscope at a high power of into 400. The magnification of the light microscope which they are talking about in the question is into 400, right? And they are saying that which organelles of the cells are clearly visible. So how we can solve this question? We know that the light microscope can view the objects that are 200 nanometers or above, right? We have studied it in the lectures of cell structure, that the light microscopes can view the objects which are of 200 nanometer or above. It is its resolution. So in order to solve this question, they are saying that whether ribosomes can be visible or not, whether endoplasmic reticulum can be visible or not. So in order to basically solve this question, what you have to memorize is that you should know the size of the ribosome, the centrioles, chloroplast, endoplasmic reticulum. Like, you need to know the size of all of these organelles. So let's discuss the size of the organelles, which is the ribosome. Ribosome has 25 nanometers size, right? Ribosome is of 25 nanometer, and then the endoplasmic reticulum is of 50 nanometers, centrioles is of 100 nanometers, and the chloroplast is of 3000 to 10,000 nanometers, right? All of these are in nanometers. Now you guys tell me, that is ribosome visible with the light microscope or not? No, it is not visible, right? Why it is not visible? Because it is of 25 nanometer, and light microscope can only view the objects with 200 nanometer or above. Similarly, endoplasmic reticulum can also not be visible. Centrioles can also not be visible. Chloroplast have its range of above 200 nanometer, which is 3000 to 10,000 nanometers. So yes, chloroplast can be visible with the light microscope. Hence the correct answer to this option is option D. I hope that this MCQ is clear to all of you. Now let's move on to question number 2 over here. Question number 2 over here says that microvilli and root hairs are characteristic structures of some cell types. Alright, we know that root hair cells is somewhat like this, right? This is somewhat the structure of the root hair cells. Then they are saying that which row, and the microvilli, if we talk about microvilli, so we know that in the intestine we have the structure known as the villi, right? And inside the villi, there in its epithelium, it is made up of the cells which are having the microvilli, right? These are the microvilli. Now they are saying that which row identifies some features of a microvillus and a root hair. Now basically they are saying surrounded by the cell wall. So we all know that root hair is surrounded by a cell wall, right? Because it is in the plant cell, and we know that it is surrounded by the cell wall, present more than one on a cell, right? More than one on a single cell. If you talk about this structure which I have drawn over here, in the epithelium of the villi, we can see the microvillus cells which are more than one on one cells, right? These are the microvilli which are more than one on one cell. Similarly, if we talk about the root hair cells, so root hair cells has only one extension, right? So the correct answer to this option would be option the correct answer to this question should be option A, right? I hope that this is clear to all of you. Now let's move on to question number 3 over here. Question number 3 over here says that the photomicrograph is of a plant cell. The cell is 25 nanometer in the width from X to Y. Alright, so they are basically telling you the actual length. The length which are basically given in the question is mostly the actual length, right? What is this? This is the actual length, right? Now they are saying that what is the magnification of the photomicrograph. We know that the formula for the magnification is equal to image length divided by the actual length, right? Now they are saying over here that we have to calculate the magnification. Alright, you have to find this. So for it you have to calculate the image length. The actual length is given to you in the question, which is 25 micrometers. Now you should also convert the image length which you are going to calculate into the micrometers. So what you will do, that you will take a scale and you will measure this length of x to y, right? Whatever the length is coming, my length, if I solve it on the paper, so my length was coming to be 4.5 centimeters, right? Now you have to convert this 4.5 centimeters into meters. You know that whenever we go from centimeter to meter, we divide the value by 100, right? So the value will come in meters. Whatever the value comes in meters, you will multiply it by 1000 and you will convert it into millimeters, and then you will further multiply it by 1000 to convert it into micrometers. Then comes the nanometer, then comes the milli, micro, nano, picometers, right? And over here also this cycle goes, but we have to only calculate it till the micrometer, right? We have to get our value in the micrometer, so our answer would come to be 45,000, right? It will come to be 45,000. So we are going to solve it in the calculator, and the answer which we will get is 2 into 10 raised to the power 3, right? So I hope that this MCQ is clear to all of you. So now let's have a look at question 4. It says that which eukaryotic cell structures all contain nucleic acids. If we talk about nucleic acid, we know the two type of nucleic acid, which are the DNA and which are the RNA. Right now RNA is of several types, which is the mRNA, right, transverse RNA, and then there comes the ribosomal RNA, right? So they are saying that which eukaryotic cell structures contain the nucleic acid. We know that the Golgi bodies do not contain any nucleic acid, so option A has been eliminated, right? Then we know that centrioles do not contain any nucleic acid, so it is also eliminated. Then over here, if we talk about over here also, centrioles do not contain the nucleic acid, so this option is also excluded, right? Now in option D, they are saying that chloroplast, mitochondria, cytoplasm and ribosomes. Now in the chloroplast, we know that yes, chloroplasts do contain the nucleic acid, as well as mitochondria also contain the nucleic acid. They have the circular DNA, right? They have the circular DNA. If we talk about the structure of the chloroplast and the mitochondria, we have studied it over there that they are having the circular DNA. Then similarly all the nucleic acid is obviously contained within the cytoplasm of the cell. And then the ribosomes, definitely ribosomes are made up of the protein plus ribonucleic acid, ribosomal RNA, right? Ribosomal RNA, ribosomal ribonucleic acid, right? So yes, option D is our correct answer over here. I hope that this MCQ makes sense and is clear to all of you. Now let's move on to question number 5 over here. Says that which statement about the viruses are correct. Now in statement 1, they are saying they contain DNA or RNA. Yes, it is correct. Plus it also contains a protein coat, right? Protein coat basically surrounds the DNA or the RNA. Statement 2 says they use the host cells to synthesize the virus protein. Yes, this statement is absolutely correct. That's why the viruses are basically known as intracellular parasites, because they cannot survive independently. They can only survive and they can only reproduce inside the human cell, inside a living cell, sorry, right? And yeah, so that's why they are called the intracellular parasites. And yes, this statement is also correct that they use the host cells to synthesize the virus proteins, right? Now in statement 3, they are saying that they have a protective coat of peptidoglycan. No, they cannot have a protective coat of peptidoglycan. This is absolutely incorrect. So the correct answer to this question is option B, 1 and 2 only. Let's now have a look at question number 6 over here. Question number 6 over here says that what are the features of the prokaryotic cells, chloroplast and the mitochondria, right? So they are saying that ribosome smaller than the 70s ribosome, 80s ribosome. So yes, this is correct. Both the chloroplast and the mitochondria contain the 70s ribosomes, right? They both contain the 70s ribosomes. Now the circular DNA. Yes, we know that chloroplast also has a circular DNA and mitochondria also has a circular DNA. Now surrounded by the double membrane. We know that the chloroplast and the mitochondria are surrounded by the double membrane, but in the question they are saying that what are the features of the prokaryotic cells, chloroplast and mitochondria. In the prokaryotic cell there is no double membrane bound organelles, right? So the correct answer for this question would be option B. Question number 7 over here says that a student used Benedict solution to test a sample known to contain the carbohydrates. At the end of the test, the solution was blue. Which carbohydrate could be present in the sample? So the Benedict test is basically used to identify the reducing sugars, right? And the color of the Benedict solution is blue at the start of the experiment. And if the reducing sugars are present, if in case the reducing sugar are present in the in the solution, then the color may change from red, orange, yellow, from green to yellow to orange to green, depending upon the concentration of the reducing sugar, right? And if there is no reducing sugar present, then the color of the Benedict would remain blue. And over here they are saying that the color of the Benedict is remained blue. So we know that, so we can clearly conclude that there was no reducing sugar present in the sample, which means that we have to look at the option and search for the non-reducing sugar. And here I can see a non-reducing sugar which is known as the sucrose. We know that sucrose is a non-reducing sugar, so hence the answer to question number 7 is option D. Now let's have a look at question number 8 over here. Question number 8 over here says that sugars with a ring structure can also have a linear structure. Alright. So which sugar molecule could be represented by the linear structure in the diagram? So they are giving us the options ribose, deoxyribose or the glucose. We know that ribose is a 5 carbon sugar. We know that the deoxyribose is also a 5 carbon sugar and glucose is a 6 carbon sugar. So let's count the number of carbons given in the structure: 1, 2, 3, 4, 5, 6. So it can be clearly concluded that it is a 6 carbon sugar. So it can be none other than the glucose only. So hence the correct answer to this question would be option D, 3 only, which is glucose. So I hope that this MCQ is clear to all of you. Let's move on to question number 9 over here. Question number 9 over here says that which row matches each molecule to a type of the bond that is present. If we talk about the ester bond, which is C double bond OO, we know that it is made in the lipids, right? Lipids, or you can say the triglycerides, right? Triglycerides. It is also present in the phospholipids. If we talk about the disulfide bond, so we know the structure of the antibody. Antibodies contain the 2 heavy chains, right, and 2 light chains, and these chains are connected with each other. The 2 heavy chains are basically connected with each other by the disulfide bond. So we know that the disulfide bond is present in the antibody. So hence the answer should be option D. So this is how we can conclude our answer for question number 10 over here is about that which row shows the features of a carbohydrate polymer which is found inside the animal cells. So we know that the carbohydrate polymer in the animal cells is the most popular one, the glycogen, right? And we know that glycogen is a branch structure. What its structure look like? If I just draw a rough diagram, so it somewhat looks like this, right? And then there is, as it is a branch structure, so it also has a branch, right? Somewhat like this is the structure of the glycogen. So they are saying that the shape of the molecule, we know that the shape of the glycogen is branch and it is more branch than the starch. It is a the comparison can also be asked in the theory questions, that what is the difference between the glycogen and the starch. So glycogen is more branch than the starch molecules, right? So then they are saying that alpha 1,4 glycosidic bond is present. Yes, we can see over here that alpha 1,4 glycosidic bond is also present and alpha 1,6 glycosidic bond is also present. So hence the answer over here would be option A, right? Option A would be our correct answer over here. I hope that this MCQ is clear to all of you. Now let's move on to question number 11. Question number 11 is stating that the statements describe the structure of the polysaccharides found in the cell walls of a certain plant. Alright, so it is the cellulose. They are talking about the cellulose because cellulose is a polysaccharide which is found in the cell walls of the certain plants. Now they are saying that which statements is true for the cellulose. Now they are asking about the correct statement. So we have to first of all read the statements and then we have to conclude that which statement is correct. Statement 1 says the polysaccharide is composed of two different monosaccharides. No, this is absolutely incorrect. If I talk about the cellulose, so it is basically a beta glucose monomer, right? It is a monomer of beta glucose. Then it says that the monosaccharides are joined by 1,4 glycosidic linkage. Absolutely correct. Beta 1,4 glycosidic linkage is used for linking the beta glucose units in the structure of the cellulose. Then in the statement 3 they are saying that polysaccharide contains pentose sugar. Absolutely incorrect, right? It contains a hexose sugar, right? It contains a hexose sugar. So hence the answer to this question is option D, 2 only. Only one statement given is correct. Now let's do question number 12 over here. Question number 12 over here is about lipids. I believe it says that the general formula for the saturated fatty acids is CnH2nO2. Now if I talk about the saturated fatty acids, so saturated fatty acids basically are the fatty acids which contain carbon-carbon single bond, right? Which of these fatty acids are unsaturated? Unsaturated fatty acids contain carbon-carbon double bond. So we can apply a trick over here. Which sort of trick we can apply over here? That we can now calculate this, we can now count the number of the atoms, right? And we can put it in one place. Like for example, for 1 if we do, so it will come to be C11, right? We will add both the C's and then hydrogen will become 20 and O will become 2. Now you guys check that whether it is satisfying the saturated fatty acid formula or not. We can clearly see that it is not satisfying the saturated fatty acid formula, right? If the C is 11, which means that if the N is 11, then the H should be 22, but it is coming to be 20, which means that it is not satisfying the saturated fatty acids formula. So it means that it is a unsaturated fatty acid. So it is also correct. Similarly, we will do it for the other three as well. For it will be C16H32 and O2, right? For C. Now for 3 it should be C18H32 and O2. For 4 it should be C19H33 O2, right? Now we know that 2 is satisfying the above equation, which is for the saturated fatty acid. So this is incorrect. 3 is not satisfying, so it is correct. And 4 is also not satisfying the above equation for the saturated fatty acid, so it is also correct. So hence the correct answer for this question is coming to be option C, which is 1, 3 and 4. Right, so it was more of a logical question. Now let's move on to question number 13, right? Question number 13 over here is saying that what enables the triglycerides to perform their functions in the living organism? Triglycerides means that they are talking about the lipids, right? That triglycerides have hydrophobic and hydrophilic regions. No, it is not correct. It only contains the hydrophobic regions. It do not contain the hydrophilic regions. Right? How? Because lipids are majorly classified as the non-polar biological molecules. Right? So the statement 1 is incorrect. Now let's look at statement 2 over here. Statement 2 over here says that triglycerides have a high ratio of carbon hydrogen bonds to the carbon atoms. Right? Yes, this is a correct statement. That is why the triglycerides have more energy than the carbohydrates. Right? This is a very popular reason. Then statement 3 says that hydrolysis of triglycerides release metabolic water. No, this is absolutely incorrect. In the hydrolysis, we give water. In the hydrolysis, the water is added. Right? We add water in the hydrolysis. In the condensation, what happens is that there is a release of metabolic water. But in the hydrolysis, we add the water. Right? So the correct answer to this question is option D, 2 only. Alright, so now let's move on to question number 14 over here. Question number 14 over here says that the diagram shows 3 examples of different bonds. So this is the bond 1, this is bond 2, and this is bond 3. Then they are saying that which bond can hold the quaternary structure of the proteins together. Alright, so we know that the bond 1 is between the hydrogen and oxygen. So it must be the hydrogen bond, obviously. It is the hydrogen bond. Then it is the peptide bond. Bond 2 is the peptide bond. And then bond C, which is between the two sulfur atoms, it is known as the disulfide bond. Right? It is the disulfide bond. Now they are saying that in the question, which bonds can hold. They are saying which bonds can hold the quaternary structures of the protein together. They are not saying that present. They are not saying the bonds which are present. So the bonds which hold the quaternary structure of the protein together are the bond 1 and the bond 3. Peptide bond is no doubt present within the proteins in the quaternary structure of the protein, but they don't play a specific role. Yeah, they don't play a major role in holding the quaternary structure of the proteins together. Right? So the correct answer to this MCQ would be option C, 1 and 3 only. Alright, so now let's move on to question number 15 over here. Question number 15 over here says that which row correctly describes the primary structure, secondary and the tertiary structure and the quaternary structure of some proteins. So let's look at the options one by one. It determines the folding of the polypeptide chain. No, I don't think so. This is correct statement. It does not determine the folding of a polypeptide chain. It is just the primary sequence of the amino acids. Right? That what is the sequence of the amino acids. The arrangement of the amino acids is basically the primary structure. The folding is most probably determined by the secondary structure. Right? Defines the order of amino acids in a polypeptide chain. Absolutely correct. Involves covalent bonds only. Yes, the covalent bonds, the peptide bonds, you can say, are the covalent bonds. So yes, it is correct. Involves peptide bond between the side chains of the amino acid. No, side chains of the amino acids are the R group. Right? So they do not involve the peptide bond between the side chains. Rather, they forms the bond between the peptide bond is formed between the amine group and the carboxyl group of the amino acid. Right? So this statement is also incorrect over here. So we can simply eliminate two options over here. One option is D. We can eliminate D over here. We can eliminate A over here. Now let's look upon option B and option C, that what is our correct answer. Right? Now over here it says usually forms immediately after the polypeptide synthesis. Can be or cannot be correct. Let's look at the further as well. If you get confused in any one of it, you can see the other two columns as well. Right? So we will look at the other two columns as well before concluding. Involves the interaction between H and O. Yes, it is correct, because you know that the hydrogen bonds are present in the secondary structure. So yes, hydrogen bonds are formed between the H and O. So yes, it is correct. Now let's look at the tertiary structure 1. It holds together by all type of bondings that occurs in the proteins. Alright, so it is saying that held together, they are not held together by the peptide bonds, right? And peptide bonds also occur in the protein, right? So this statement is incorrect. They are not held together by the peptide bond. In the statement they are saying it is held together by all type of bonding that occurs in the protein, so it is incorrect, right? So we can simply eliminate option B as well. Now let's look at option C, which is our correct answer. No doubt it is essential for the function of the enzymes and receptors. Yes, we know that enzymes have a tertiary structure. And if I talk about the quaternary structure, yes, it is formed when two or more polypeptide chains join together. So hence the correct answer to this MCQ is option C, right? I hope that this MCQ is clear to all of you. It was a hard MCQ, no doubt, but I hope that it makes sense and it is clear to all of you. So let's look at question number 16 over here. It says that which relationships could be investigated using a colorimeter. If I talk about the colorimeter, what is a colorimeter? It is an instrument which is used to measure the absorbance or the transmission of the light by a solution, right? You will simply place a solution and you will strike a light on it and you will measure the absorbance or the transmission of a light by a solution, right? So what it will do, what is the benefit of it? It is that it is used to quantify changes in the color intensity in a solution, right? It will help you to quantify the changes in the color intensity in a solution. Now let's read the statements one by one and let us see that what could be investigated using a colorimeter. The effect of light intensity on the rate at which the solution of the light sensitive dye changes from green to colorless. So they are basically saying that whenever you strike the light on it, so the rate at which the solution which you have put in the beaker of a light sensitive dye changes from green to colorless. You have to calculate the basically they are saying that can the rate be calculated uh, from the solution at which the color changes from the green to colorless, right? So yes, it can be calculated using a colorimeter because the change in the dye's color intensity can be measured. So it can be measured, so it is correct. In the statement 2, they are saying the effect of the temperature on the rate of the breakdown of the cell membrane in the tissues with pigmented cells such as beetroot, which is the red beet. Yes, so this can also be investigated using the colorimeter. Why? Because of the leakage of the pigment from the cells, right? The pigment, because we know that it is a pigmented cell, right? So pigments are colorful and it is also red beet. So as the pigment will leak out from the cell into the surrounding solution, so there will be a change in the color intensity. So yes, it can be investigated, right? The effect of pH on the rate of the release of oxygen from the breakdown of hydrogen peroxide by catalase. No, this cannot be calculated because oxygen release do not involve a color change, right? They are saying that the oxygen is released, so it do not involve a color change, right? Colorimeter can be only used to investigate about the things which give a color change, right? So yes, in the statement 4, they are saying the effect of the light intensity on the rate of change of skin color of the lizard that becomes paler in the brighter light. If I think on it, so this can also not be investigated using a colorimeter. Why? Because the color change occurs on the lizard skin, right? It is not occurring in a solution. In the colorimeter, we are talking about the solution specifically, right? Absorbance or transmission in the solution, right? So this is also incorrect. This statement is also not correct. So the correct answer for this MCQ would be option B, 1 and 2 only. I hope that it makes sense. Question number 17 says that which transport mechanism does not require a concentration gradient to be present in order to take place. All of the other three, facilitated diffusion, osmosis and transpiration, requires a concentration gradient. However, exocytosis, which is the process in which the material exits the cell or the vesicles exits the cell, and releases its content out of the cell. So that process is known as the exocytosis, which is the release of the material outside the cell. It does not require any concentration gradient, right? So the correct answer is for question number 17 is option A. Now let's look at question number 18 over here. Question number 18 over here says that high concentration of ethanol disrupts the cell membrane structure by denaturing the proteins and increasing the separation of adjacent phospholipid molecules. As a result, cell membranes can decrease in thickness by up to 30% and become more permeable. So basically you have to first understand that what ethanol is basically doing. It is denaturing the proteins in the cell membrane, right? It is increasing the separation of the adjacent phospholipid molecule, which means that ethanol is making the cell membrane more permeable, right? Over here it is also written that it is making the cell membrane more permeable. So then they are saying in the question that yeast cells releases ethanol as a waste product in the metabolism in response to increased ethanol concentration in the environment. Yeast cells are able to increase the tolerance of their cell membranes to ethanol. Now further in the question they are saying that which statement correctly explains a response to ethanol that could account for the increase in the tolerance of yeast cell membrane to ethanol, right? So they are saying that the response of the ethanol is what? It is to make the cell membrane more permeable, right? Now they are saying that which statement is basically increasing a response to ethanol that could account for the increase in the tolerance of yeast cell membrane to ethanol, right? Means that there should be a condition which is making the yeast cell membrane less permeable, right? Less permeable, then only the tolerance will be developed, right? Because what the ethanol is doing, it is making the cell membrane more permeable. So for the resistance you have to make for the tolerance you have to make the yeast cell membrane less permeable. Alright, so now let's discuss option A. It says that decreasing the ratio. It says that Alright, so now let's discuss option A. It says that decreasing the ratio of saturated fatty acids to the unsaturated fatty acids within the cell membranes helps to prevent the tails of the phospholipids on one side of the bilayer from sliding past the tails of the phospholipids on the other side of the bilayer, which means that it is becoming less permeable, right? It is not disrupting the structure very much. Now what does it means when they talk about that the ratio to the saturated fatty acid and the unsaturated fatty acid, it decreases, right? If the ratio to the saturated and unsaturated fatty acid basically decreases, it means that there are more unsaturated fatty acid, right? And there is less saturated fatty acids. Alright, and if I talk about the saturated fatty acids are not having the kinks, and if I talk about the unsaturated fatty acids, so the unsaturated fatty acids have the kink, right? And they are saying in the question which explains a response to ethanol that could account for an increase in the tolerance of yeast cell membrane to ethanol, right? Ethanol is doing, it is making the membrane too permeable, right? And it is disrupting the structure of the membrane, and we are talking about the tolerance. We have to give the correct answer in account for the tolerance, right? So now if I talk about the unsaturated fatty acids, you can see over here that as the ratio decreases, so the number of the unsaturated fatty acid basically increases, right? So now because as the fatty acids are as the phospholipid molecules, the unsaturated fatty acids are having the kinks in them, right? They are having the kinks in them, so it will prevent the phospholipids from packing too closely, right? Because of these kinks over here, it will prevent the phospholipid molecules from packing too closely. So this will prevent the excess fluidity, ensuring the membrane remains stable even when exposed to ethanol. So hence the correct answer to this is option A, because over here they are saying that the membrane fluidity will decrease because of the more unsaturated fatty acids over there, right? So yes, the correct answer for question number 18 is option A. I hope that this is clear to all of you. If I discuss the other options, that why other options are incorrect, so let's see, let's read other options as well, and let's conclude that why these options are incorrect. Increasing the proportion of palmitoleic acid, a C16 unsaturated fatty acid, to oleic acid, a C18 unsaturated fatty acid, in the phospholipids of the bilayer increases the fluidity of the cell membrane. So no, this option is incorrect. Why? Because yeast need to reduce, right? Yeast needs to reduce the fluidity, not increase the fluidity of the membrane, right? Ethanol is already increasing the membrane fluidity, so this option is incorrect. Now if I talk about option C over here, so activating a cell signaling pathway triggers the unfolded protein response to the endoplasmic reticulum. The unfolded protein response causes the protein synthesis and initiates the cell death in yeast cells with a high proportion of misfolded proteins. Now basically they are talking about the UPR, which is the unfolded protein response, right? It deals with the misfolded proteins in the endoplasmic reticulum first of all, ठीक है, not in the endoplasmic reticulum, not the membrane stability, right? And the other thing is that cell death does not increase the ethanol tolerance. It removes the damaged cell but does not help yeast to survive ethanol stress, right? So this is also incorrect. Now if we talk about the option D over here, right? Option D is saying increasing the proportion of ergosterol in the cell membrane prevents the accumulation of polar molecules such as ethanol within the cell. Ergosterol in yeast cells has a similar effect on the membrane permeability as cholesterol in the mammalian cell. So basically, ergosterol stabilizes the membrane similar to cholesterol in the animal cell. This is correct, but however, the yeast cell usually decreases the ergosterol levels in response to the ethanol stress, right? Why? Because too much ergosterol makes the membrane rigid, which ethanol can still penetrate, right? So hence this statement is also incorrect. So I hope this was a very difficult MCQ. I hope that now this MCQ is clear to all of you. Alright, so now let's move on to question 19 over here, and let's see what they are asking us in question 19. It says that in an investigation, a plant cell was placed in a pure water, right? Acha, pure water, if they talk about the pure water, so it means that the pure water has the highest water potential, right? If you are talking about the pure water, so it has the highest water potential. Then they say that the initial rate at which the water molecules entered the cell R was greater than the initial rate at which the water molecules left the cell. Then over here in the next point, they are saying that in the second investigation, a plant cell of the same type was placed in a solution with a water potential equal to that of the cell content. What will happen to the second investigation over a period of 5 minutes? Alright, so basically what is happening over here, that in the first investigation, right, in the first investigation, which was this, what happens is that the plant was placed in a pure water and the pure water was having the highest water potential, right? So this caused the net water uptake, right? What it does is it causes a net water uptake, right? So which means that more water entered the cell than the one which is leaving the cell, which means that there is the net movement of water inside the cell, right, resulting in the initial rate of water entry R, right, in resulting in the initial rate at which the water molecules entered the cell R, right? And then what happens that in the second investigation, they are saying that plant cell was placed in a solution with the same water potential as the cell content. So they are saying that in option A, they are saying that water molecules will not enter or leave the cell because the water potential of the cell content is equal to that of the solution. So no, this statement is incorrect. Why? Because water molecules are going to enter and leave the cell, but there will be no net movement of water, right? There will be no net movement of water, but the water molecules will enter or leave the cell, right? So I hope that you guys are understanding it, that there will be no net movement of water inside and out of the cell, but there will be a movement of water in the equal rates inside and out of the cell, right? If I look upon the option B over here, so option B over here says that water molecules will enter and leave the cells in equal amount. Yes, this is correct, right? And then both at an initial rate that is less than r in the first investigation. Yes, this is correct, because in the first that water molecules will enter and leave the cell in equal amount. Yes, this is correct, because there will be no net movement of water, right? Because why? Because it is placed in a solution with a water potential equal to that of the cell content, so there will be no net movement of water, but there will be the movement of water into and out of the cell. Random movement will be there. Then they are over here saying both at an initial rate that is less than r in the first investigation. Obviously in the first investigation what was happening, that the pure water was used, right? And pure water has the highest water potential, which means that it had a steep water potential gradient, right? It has a steep, or you can say that it has a strong water potential gradient, which drove the rapid water movement into the cell, right? Which drove the rapid water movement into the cell. Over here the water, and over here what is happening in the second investigation, that the water is entering and out of the cell only due to the random movement and at a very lower rate. Why? Because there is no water potential gradient over here. The water potential gradient is the same, and there is no net movement of water as well. So hence the correct answer for question 19 would be option B. I hope that this MCQ is clear to all of you. Now let's move on to question number 20. Question 20 over here says that the protein P53 is produced in a cell in response to DNA damage. This protein stops the cell cycle for a short time just before the DNA is replicated so that the DNA can be repaired. At which phase of the cell cycle will this stop occurs? Acha, basically if we talk about the cell cycle, so it is basically divided into G1 phase, then comes the S phase, then comes the G2 phase, right? And then comes the whole phase of the mitosis, if I am talking about the mitotic cell cycle, mitosis, right? And mitosis may we have the prophase, metaphase, anaphase, telophase, right? And then cytokinesis happens. Now in the G1 phase it is the growth phase. Then in the S phase is basically a phase where the DNA replication occurs, right? S phase is a phase where the DNA replication generally occurs, and they are saying that this protein stops the cell cycle for a short time just before the DNA is replicated. So DNA is replicated in the S phase. So just before DNA replication would be the G1 phase. So it is saying that which phase of the cell cycle will this stop occur. So the correct answer to this MCQ is option B, which is the G1 phase, right? So hence option B is our correct answer.